The overall goal of this preclinical platform is to provide a standard procedure to identify and characterize the mechanisms driving acquired resistance to targeted therapies.
This method can help to implement genome-driven oncology, in particular address key questions about tumor heterogeneity and the multiple mechanisms responsible for acquired resistance to targeted therapy. The main advantage of this technique is the time and cost-effective approach, which allows the in vitro investigation of resistance mechanisms to targeted therapy.
The implication of this technique extends to cancer therapy, as drug resistance is still challenging for emerging targeted therapies.
This method can provide insight into the mechanism involved in targeted therapy resistance, but it can also be used to study the inefficacy of conventional chemotherapeutic agents.
Generally, operators new to this method will struggle with the generation of drug-resistant subclones, the step most difficult to standardize.
We first conceived the idea for this method when we characterized the acquired resistant mechanism to trastuzumab in HER-positive gastric cancer cell lines.
Visual demonstration of this method is critical, as several techniques are needed for the generation and validation of in vitro drug-resistant models, including restoring sensitivity by gene knockdown.
To begin this procedure, seed NCI N87 cells into 25 square centimeter culture flasks. Add 10 micrograms per milliliter of trastuzumab to the culture medium in each flask. Expose the cells to this starting dose until they resume growing. After cell growth has resumed in approximately 2 weeks, expose cells to a 100 microgram per milliliter concentration dose of trastuzumab. Continue gradually increasing the trastuzumab concentration, waiting until the cells resume growth before each exposure, until a concentration of 400 micrograms per milliliter is reached.
Cell line might exhibit different sensitivity to different drugs. One solution to this problem is to expose cells to gradually increasing drug doses, starting from a dose about 10-fold lower the plasma peak concentration, until cells keep growing.
Next, seed cells into 24 multi-well culture plates. Expose the seeded cells to increasing target therapy inhibitor concentrations from 100 micrograms per milliliter to 400 micrograms per milliliter. Incubate in a CO2 incubator at 37 degrees Celsius for 15 days. After this, remove the medium and rinse the cells with 10 milliliters of PBS1X. Remove the PBS1X and add between 2 and 3 milliliters of fixation solution. After 20 minutes, remove the fixation solution and add 2 milliliters of 0.5% crystal violet solution to each well. Incubate at room temperature for 2 hours. Then, use a pipette to carefully aspirate the crystal violet solution. Immerse the plates in tap water to rinse off any remaining residues. Air dry at room temperature for up to 2 days. Next, use an inverted microscope at 4x magnification to count colonies containing more than 50 cells. Calculate the relative resistance IC50 ratio as outlined in the text protocol. To begin, prepare a single-cell suspension by trypsinization as outlined in the text protocol. Using a hemocytometer and trypan blue staining, count the cells. Then, seed 2.5 times 10 to the 5 cells onto a T25 flask for each condition. Next, dilute 12.5 microliters of each gene-targeting siRNA oligonucleotide or negative control siRNA oligonucleotide in minimal essential transfection medium. Add a cationic liposome transfection reagent at a 1-to-1 ratio with the oligonucleotides. Incubate at room temperature for 20 minutes. After this, add 700 microliters of the transfection mix to each T25 flask. Incubate at 37 degrees Celsius for 1 to 3 days. use RT-PCR and Western blot analysis to verify gene knockdown. Next, seed cells into 24 multi-well culture plates. Expose the cells to increasing concentrations of target therapy inhibitor from 100 micrograms per milliliter to 400 micrograms per milliliter.
For the efficacy of gene knockdown, certain oligonucleotide must be specific for reducing target expression. Design tools and a mixture of siRNAs for the same target gene are useful for this purpose.
Incubate in a CO2 incubator at 37 degrees Celsius for 15 days. Remove the medium and rinse the cells with 10 milliliters of PBS-1X. After removing the PBS, add 2 to 3 milliliters of fixation solution. Then, remove the fixation solution and add 0.5% crystal violet solution. Incubate at room temperature for 2 hours. Using a pipette, carefully aspirate the crystal violet solution. Next, immerse the plates in tap water. Air dry at room temperature for up to 2 days. After this, use an inverted microscope at 4x magnification to count the colonies containing more than 50 cells. In this study, 3 gastric cancer cell lines, which express a high level of HER2 and are sensitive to trastuzumab, are grown in culture medium containing the targeted therapeutic agent. From this, 3 subclones, characterized by a stable resistance phenotype, are obtained. Of the subclones, HR-NCI-NH7 is seen to be the most resistant, reaching a relative resistance IC50 of 28.57. Analysis of protein and gene expression supports the hypothesis that the gene IQ-GAP1 plays a pivotal role in the resistance phenotype of the HR-NCI-N87 subline. As shown here, IQ-GAP1 protein level is 5-fold higher in the resistant subclone than in the parental cells, and IQ-GAP1 gene expression is 2.5-fold higher in the HR-NCI-N87 subline than in the original subclone. IQ-GAP1 gene silencing is then performed to evaluate its role in the development of the trastuzumab resistance phenotype. A clonogenic assay further confirms the hypothesis of IQ-GAP's importance. As seen here, the gene silencing restores the cell's sensitivity to trastuzumab, lowering the IC50 value to 7 micrograms per milliliter.
While attempting this procedure, it is important to keep in mind that it is not generally easy to obtain drug-resistant cell subclones, and that this method holds a potential failure risk. Through this procedure, it is possible to obtain several cell subclones characterized by different mechanisms driving acquired resistance to target therapies. The development of standard procedure for the in vitro modeling of target therapy resistance should allow oncology researchers to investigate novel druggable targets to overcome drug resistance, which is greatest criticism of the latest generation therapies.